Welcome to the Module 1, Session 1 review of our Introduction to Epidemiology course. This session focuses on the foundational figures and concepts shaping the field of epidemiology. Let's quickly review the important concepts from this session, making sure that we have thoroughly covered our session and learning objectives. This module is designed to provide you with a solid foundation in epidemiology, touching upon its definition, historical significance, and the profound impact it has on public health and medical practices. As we review these objectives, reflect on how each concept contributes to the bigger picture of disease control and public health improvement. For our first learning objective, 1.1, we focus on defining epidemiology. We adopted the CDC's definition, which describes epidemiology as the study of distribution and determinants of health-related states among specified populations, and the application of that study to the control of health problems. This definition underscores the discipline's crucial role in understanding health patterns, risks, and outcomes in different populations. But how does epidemiology fit within the broader scope of evidence-based medicine, or EBM? To answer this, we turn to David Sackett's definition of EBM, which integrates clinical expertise, patient values and their preferences, and the best research evidence published in the literature. As illustrated in our diagram, these three components converge to form the core of evidence-based medicine. Notice how epidemiology, along with biostatistics and clinical study design, is essential in developing and understanding the best research evidence. This evidence is a cornerstone of EBM, informing clinical decisions and patient care. In essence, epidemiology provides the tools and methods to generate and interpret the research evidence. It equips healthcare professionals with the data needed to evaluate the effectiveness of interventions, understand disease patterns, and ultimately improve patient outcomes. Through this lens, we can appreciate epidemiology's integral role in shaping evidence-based practice, influencing local and global healthcare strategies, and guiding public health policies. As we move into our second learning objective, 1.2, we delve into the groundbreaking work of John Snow, Ignaz Semmelweis, Louis Pasteur, and Robert Koch. These pioneers were instrumental in challenging and debunking the disease theories of their era, significantly contributing to the development of germ theory. Let's look at one of John Snow's initial investigative efforts. The image shown is of an 1850s street scene in London. Observe the layout of the street. On the top of the image, label 1, we see sewage flowing towards the neighborhood's public water pump. In contrast, on the bottom side, labeled 2, the sewage is flowing away from the water source. Through meticulous data collection and analysis, Snow identified a crucial pattern. The incidence of cholera deaths was significantly higher on the side of the street where the sewage contaminated the water pump flowing towards it. This correlation led to further data collection efforts, strengthening his theory that contaminated water was the source of the cholera epidemic. Another piece of evidence John Snow used was the comparison of how London residents received their water sources. This slide illustrates the positions of two major water companies in London during the mid-19th century, the Southwark and the Vauxhall Water Company, shown in blue, and the Lambeth Water Company, depicted in red. Initially, both companies drew their water source from the Thames River intakes downstream from where the river was heavily contaminated with sewage. However, in 1852, for financial reasons, the Lambeth Company made a significant change. They relocated their water intake to a site upstream in the Thames, away from the primary sources of sewage contamination, as shown in the red on the bottom of our map. Snow sees this opportunity as a natural experiment, he meticulously collected and compared cholera mortality rates among populations served by these two companies, both before and after relocation of the Lambeth Company's intake. What he found was striking. The data revealed that prior to 1852, when both companies drew water from contaminated sections of the Thames, cholera cases were high among their consumers. However, after 1852, when the Lambeth Company moved upstream, the rates of cholera significantly decreased among the consumers compared to those receiving water from the Southwark Vauxhall Company, which continued to draw water from the contaminated part of the Thames. This comparison compiled more evidence supporting Snow's hypothesis that cholera was being transmitted through contaminated water. Snow's next crucial analytical tool was using site maps in the Soho district in London during the cholera outbreak. As you can see, one of his original maps on the bottom right. These maps were far more than ordinary diagrams. They were essential instruments in epidemiological detective work. On the left, you can see a color-enhanced version of the John Snow's original site map to help visualize the data. The map on the left highlights all the water pumps in blue, including the Broad Street pump in a large blue star, and the cholera deaths in red. Each red dot on this map represents not just the data point, but a human tragedy, a life claimed by cholera meticulously plotted by Snow in his quest to unravel the mystery of the outbreak. The most striking aspect of these maps is the apparent clustering of cases in proximity to the Broad Street water pump. This dense accumulation of cases around the pump emerged as crucial evidence pointing to a common source of contamination. Snow's mapping and analysis revealed that residents who used the Broad Street pump experienced significantly higher rates of cholera. This vital correlation shifted the focus of his investigation towards this pump as the source of contamination, leading to the eventual removal of the Broad Street pump handle. Snow's map directly challenged the miasma theory, proposing instead that the waterborne nature of the disease was to blame. The use of geographic mapping in this public health crisis underscores the immense power of using data in epidemiology. It stands as a testament to the way careful observation and systematic data collection can lead to groundbreaking discoveries, significantly enhancing our understanding and control of infectious diseases. Even though the transformative work of Ignaz Semmelweis on puerperal fever in hand washing preceded John Snow's work, its impact on germ theory wasn't recognized as important until years later. On the left, you see a graph depicting the mortality rates from puerperal fever at the Vienna General Hospitals from 1841 to 1846. Observe the marked difference in the rates between the two wards. The first clinic, shown in pink, was attended by medical students and doctors, while the second, in blue, was staffed by midwives. The higher mortality rates in the first clinic caught Semmelweis's attention, leading him to hypothesize that particles from the cadavers of the autopsies performed by medical students and physicians were being transmitted to their patients, contrary to the prevailing miasma theory of disease. To test this hypothesis, Semmelweis implemented a protocol requiring that all medical students and physicians wash their hands after performing autopsies. Now turn your attention to the graph on the right. It marks an important finding in medical history, where we can compare the longitudinal rates of infection before and after the introduction of hand washing. The implementation of hand washing led to a dramatic decline in mortality rates in the first clinic. This stark reduction, which is clearly delineated by the red line on the graph, and the drop in mortality rates right after, provided compelling evidence that hand hygiene is critical in preventing the transmission of puerperal fever. We now turn to one of Louis Pasteur's most significant experiments, which utilizes the iconic swan neck flask, a cornerstone in the development of germ theory. The image before you encapsulates Pasteur's experimental design that was crafted to test the hypothesis of spontaneous generation. These flasks had a long, curving neck designed to trap airborne particles, preventing them from getting into the broth unless the neck was broken. Initially, these flasks held a clear broth that Pasteur had boiled to eradicate any life forms. In the top row, you can see where the swan neck flask remains intact. The broth stays clear, demonstrating that no microbial life arises spontaneously. Now moving to the middle row, we notice that when the swan necks were removed, it allowed for direct exposure of the air and the microbes in it to enter the broth and grow. In the bottom row, where the flask is tilted and the previously trapped particles that are in the curve of the swan neck meet the broth, we witness microbial growth as well, decisively demonstrating that microorganisms originated from the environment, not the broth itself. This experiment did more than just debunk the myth of spontaneous generation. It offered robust empirical support for germ theory as well, fundamentally altering our comprehension of infection and disease transmission. Pasteur's innovative work established the foundation of modern practices such as sterilization, aseptic surgery, and the broader field of microbiology. As we conclude our review session, we turn to the seminal work of Robert Koch. To the right, you'll find a visual representation of Koch's postulates, a set of methodological criteria he established in the late 19th century that set the gold standard for microbial research and pathogen identification. First, at the top of the image, notice the two animal figures, one exemplifying a diseased animal and the other a healthy animal. According to Koch's first postulate, the pathogen suspected of causing the disease must be present in all instances of the illness, but absent in a healthy organism. This postulate underscores the specificity of pathogens to their associated diseases. Moving on to the second postulate, depicted by the Petri dish cultures. Koch asserted that the microorganism must be isolated from the disease host and cultured in a pure state and not found in a healthy animal. This isolation is critical to avoid contamination and ensure accurate identification, laying the groundwork for further examination. In the third postulate, as illustrated, involves inoculating a healthy animal with the isolated microbe, which must then induce the same disease, thus establishing causal link of the disease rather than mere correlation. And finally, the fourth postulate describes that simply inducing the disease isn't sufficient. The microbe must then be re-isolated from the newly infected animal and confirmed to be the same as the original pathogen, solidifying its role as the causative agent. The profound impact of Koch's postulates on germ theory of disease is immeasurable. They provided a systematic and empirical approach to link specific diseases to specific microorganisms. Koch's work forms the bedrock of modern medical science, found profoundly influencing our grasp of infectious diseases and spearheading the development of vaccines and antibiotics.